that would be the top of a human brain because the way our bodies are built. But on this view, you don't have a ton that you can see. There's three things that you can see externally. You have the cerebral cortex, which is the majority of what you're seeing in this view. This is the folded and grooved stuff. This is where your conscious activity takes place. So your ability to constantly move. These are from sheep, so there's not a lot of conscious thought in a sheep's brain, but in a human this would be important for your personality and memory, more personality and memory, things like that. But the cerebral cortex, we can identify some landmarks on it. The bulges and folds in there have names. The bulges, where it sticks up a little bit, are called gyri, or a gyrus is the singular. And then the grooves in between them are called sulci, or sulcus is the singular. The sulci will typically be pretty obvious because they'll have dark lines where blood vessels and other material are sort of accumulated in that crevice. In a human brain, and we have some slices of human brain to look at over here, you'll see the sulci are much deeper than they are in a sheep's brain. The sulci and the gyri, they're landmarks, but they do sort of function in terms of allowing you to cram more stuff into the brain. So the deeper the grooves are, the more neurons your brain can have. And so what it comes down to is intelligence. Some of that comes from the number of neurons in your brain. And what's going on there is that in a human brain, you have lots of deep sulci and big bulges, gyri, that make you have more neurons. Sheep, obviously, their brain is pretty smooth, not the brightest animals in the barnyard. But the basic function of the gyri and the sulci is the same. It allows you to cram more neurons into a smaller space. And there are some sulci that have specific names, but the one large place that we'll see something like that is what's called the longitudinal fissure, which is typically held together by some membrane. If you gently take your fingers and sort of separate the two halves of the cerebral cortex, you should find that you can sort of poke it out with a probe and at least tear. You don't want to pull it all the way apart yet. You should be able to tear that and see this line that runs down the medial part of the brain. This separates your left and right hemispheres.
Yeah.
And there are connections deeper in that we'll expose when we cut the brain open in a little bit, but it's going to allow you to sort of isolate the different parts of the brain. So the fissure itself allows you to isolate the left and right hemispheres, and then other parts of the brain that we'll see next time, that we'll see later, allow the two parts to talk to each other. The next couple of items, 2 and 3, are inside. We'll see them in a second. The other thing that you can see very easily from this view is the cerebellum, which is this massive material that is behind the cerebral hemispheres. The cerebellum is involved in a lot of control of muscle movement and regulating accuracy of those movements. So learning how to throw a ball or ride a bike gets better with practice. Your cerebellum is part of why that's the case. And that's why damage to the cerebellum is often associated with movement disorders called ataxia, where you lose your coordination and no longer have the fine muscle control that you used to have. And then sticking off below the cerebellum, you should have at least a portion of the spinal cord visible as well. And we'll talk more about that on the other view. So everybody's seen the cerebral hemispheres, the cortex, and the spinal cord? So the spinal cord is facing up towards you. Put your thumb on the cerebellum and put your other fingers underneath on the ventral side of the brain and gently bend it down. Again, you'll find some membrane that sort of holds us in place, so you might want to pick at it with a probe or with your fingers, sort of tear some of those membranes. And when you do that, you should get a view similar to this, where to be blunt, it looks like you're being moved. Tell me it doesn't look like that. Come on. I'm not just immature. I'm immature, but I'm not just immature. So the structures that you're seeing, there's two paired structures that make up the portion that you can see right here, and there's a single structure up above them. It's a little hard to see because it's a little bit in shadow. But if you look inside there, sitting up above this area, you have a single structure kind of in the middle. That's the pineal body or the pineal gland. It's involved in producing the hormone melatonin that helps regulate your sleep-wake cycle. And this is part of the reason why you have things like jet lag. If you travel across several time zones, the amount of melatonin your body is producing is synchronized to the amount of light your eyes are seeing. And the problem is when you jump time zones, the light doesn't match up to what you were seeing. It takes a while for melatonin levels to recover. So part of what's going on with jet lag is that or even daylight savings time, it's been shown that affects you for about 2 weeks. So you feel grumpy and tired for a couple of days, but your brain function is actually off for a couple of weeks during that time period because of melatonin levels being off. Below the pineal body, you have this structure which consists of 4 lobes called the corpora quadrigemina. Corpora quadrigemina means the body with 4 parts, and that's because you have 2 upper parts and 2 lower parts, so that's the 4 parts. The 2 upper parts, the moving bits, are called the superior colliculus, and the 2 lower ones, which are a lot smaller and more just like a line, are the inferior colliculus. The job of the superior colliculus is to build what we would call your visual map. So the fact that you can look around the room and say, you know, my notebook's on my left, and my pencil's on my right, and the brain is right in front of me, whatever, that's your superior colliculus that does that for you. And this is why with babies, you give them mobiles and things sort of whap at when they're in the crib, when they're first born, because they have to build that map and understand where left and right and things like that are, and so connections in the superior colliculus are really important for that. In the inferior colliculus, you're dealing with the what's called the auditory map, or your map of sound. So when someone calls you, you say, oh, that came from my left, or it came from my right, or you know, behind me, in front of me, whatever. And these 2 things are tied together closely physically, but they're also tied together very important functionally, because you'll tend to turn and look at loud noises and things like that because they tend to be an important signal. And so by having the 2 maps right next to each other, when you hear a loud sound, the first thing you do is try to turn your head and look at it. Because as visual animals, we tend to need our eyes to be on it as quickly as possible. And so you hear a loud noise and people are all looking around trying to figure out where it came from. That's because they're trying to use these 2 maps to get their eyes pointed at whatever it was that made that noise. That's it for the dorsal side.